हेलो हेलो वेलकम टू द सुमो बायोलॉजी ऑनलाइन क्लासेस। तो हम लोगों ने चैप्टर चालू किया है सेल द यूनिट ऑफ लाइफ। वी हैव स्टार्टेड द चैप्टर नंबर 8, क्लास 11 बायोलॉजी, सेल द यूनिट ऑफ लाइफ. In the last lecture we have discussed about prokaryotic cell. In this lecture we are going to discuss eukaryotic cell. Okay, so let's begin with eukaryotic cell. Eukaryotic cells. The eukaryotes include all the protists, plants, animals and fungi. So all the protist, plants, animals and fungi are representative of eukaryotic cell. Only the kingdom Monera contains prokaryotic cell, and kingdom Monera represented by bacteria. Rest all the four kingdoms. R.H. Whitaker proposed 5 kingdom classification, and 5 kingdoms according to R.H. Whitaker was Monera, Protista, Fungi, Plantae and Animalia. So these four are representative of eukaryotes. So all the eukaryotes include all the protists, all the plants, all the animals and all fungi. In eukaryotic cell there is an extensive compartmentalization of cytoplasm through the presence of membrane bound cell organelles. In case of eukaryotic cell there is the presence of membrane bound cell organelle, and due to the presence of membrane bound cell organelle, cytoplasm get divided into compartments. So compartmentalization of cytoplasm is due to presence of membrane bound cell organelles, and membrane bound cell organelles, presence of membrane bound cell organelles are characteristic feature of eukaryotic cell. In case of prokaryotic cell, membrane bound cell organelles are not present. Eukaryotic cells possess an organized nucleus. In case of eukaryotic cell, a well-defined nucleus is present. Well-defined nucleus means genetic material bound by nuclear membrane. Whereas in case of prokaryotic cell, genetic material is not bound by nuclear membrane. So genetic material is said to be naked. In case of prokaryotic cell, we use the term naked genetic material. And histone proteins are also not present in prokaryotic cell, whereas in case of eukaryotic cell, histone proteins are present. Eukaryotic cells possess an organized nucleus with the nuclear envelope. In addition, eukaryotic cell have a variety of complex locomotory and cytoskeleton structure. In case of eukaryotic cell, you will find the presence of variety of complex locomotory and cytoskeleton structure. Their genetic material is organized into chromosome. Their genetic material means genetic material of eukaryotic cell. It is well organized into chromosome. All eukaryotic cells are not identical. All the cells are not identical to each other. Plant and animal cells are different. As the former possess cell wall, the outer membrane, outer delimiting boundary of a plant cell is rigid cell wall, whereas in case of animal cell, the outermost boundary or delimiting boundary of animal cell is plasma membrane, cell membrane. Animal cell is not bound by cell wall. Cell wall is absent in case of animal cell. So there is a difference between plant cell and animal cell. In case of plant cell wall, the outermost boundary is cell wall. Plastids and large central vacuole which are absent in animal cell, and in case of plant cell you will find the presence of large central vacuole, whereas large central vacuole is absent in animal cells, and plastids are also the presence of plastids are characteristic feature of plant cell, whereas plastids are not present in animal cell. On the other hand, animal cells have centriole, which are absent in almost all plant cells. So basic difference between plant cell and animal cell. In case of plant cell, the outermost boundary is presence of cell wall. Cell wall is present, whereas cell wall is absent in case of animal cell. Plastids are present in case of plant cell, whereas plastids are absent in case of animal cell. Okay, a large central vacuole is present in plant cell. Central vacuole is present in animal cell but not large. So large central vacuole is not present in animal cell. And presence of centriole would be present in animal cell and centriole is absent in plant cell. So these are the differences between plant cell and animal cell, and both are, plant cell and animal cell, both are the type of eukaryotic cell. So all the eukaryotic cells are not identical to each other. There is some difference between animal cell and plant cell. On the other hand, animal cells have centrioles which are absent in almost all plant cells. ओके, तो ये बात हुई कि यूकैरियोटिक सेल में आप देखेंगे वेल डिफाइंड न्यूक्लियस है, जेनेटिक मटेरियल न्यूक्लियर मेम्ब्रेन से बाउंड है, कंपार्टमेंटाइजेशन देखने को मिलेगा ड्यू टू द प्रेजेंस ऑफ मेम्ब्रेन बाउंड सेल ऑर्गेनेल्स, एंड ऑल द यूकैरियोटिक सेल्स आर नॉट आइडेंटिकल. आपको डिफरेंस देखने को मिलेगा प्लांट सेल और एनिमल सेल में. सो दिस इज द upper one is the diagram of plant cell and lower one is the diagram of animal cell. So you can see the presence of cell wall in case of plant cell. This is the cell wall. Outermost boundary is made up of cell wall. Nucleus is there, well membrane bound nucleus, nucleolus, spherical structure, Golgi body, endoplasmic reticulum. These are the endoplasmic reticulum, mitochondria, plasmodesmata. Plasmodesmata are the cytoplasmic bridge between two adjacent cell, microtubules, locomotory organelles, peroxisome. This is the cytoplasm, central vacuole. So these are the, and plastids. Where are the plastids? Plastids, cytoplasmic, endoplasmic reticulum, lysosome, endoplasmic reticulum, Golgi body, mitochondria, vacuole, cell wall, mitochondria, chloroplast. This is the plastid. Plastids are present in plant cell. This is the diagram for animal cell. Outermost boundary, cell membrane, plasma membrane, cell wall is not there. Centriole is present, centriole are absent in plant cell. This is the smooth endoplasmic reticulum, mitochondria, lysosome, rough endoplasmic reticulum, nucleus, Golgi body, microvilli. Okay. Let us now look at the individual cell organelles to understand their structure and function. Now we are going to discuss one by one individual cell organelles and understand their functions and structure. The first one is cell membrane. The detailed structure of the membrane was studied only after the advent of electron microscope in the year 1950. 1950 के दशक के बाद, 1950 के बाद when the इलेक्ट्रॉन माइक्रोस्कोप डेवलप, तो इलेक्ट्रॉन माइक्रोस्कोप के डेवलपमेंट के बाद ही इसका डिटेल स्ट्रक्चर हम लोग को पता लग पाया है. मीनवाइल केमिकल स्टडीज ऑन द सेल मेम्ब्रेन स्पेशली इन द ह्यूमन RBC इनेबल द साइंटिस्ट टू डिड्यूस द पॉसिबल स्ट्रक्चर ऑफ प्लाज्मा मेम्ब्रेन. लेकिन ऐसा नहीं है कि तब तक कुछ काम नहीं हो रहा था, साइंटिस्ट काम कर रहे थे और स्पेशली ह्यूमन RBC पे वो काम कर रहे थे, ह्यूमन RBC पे स्टडीज़ कर रहे थे. Meanwhile, chemical analysis was going on chemical studies on cell membrane, especially in the human RBC. So why human RBC is the most preferred material to study the composition of cell membrane? Because human RBC, mature human RBC is enucleated. ये क्वेश्चन आपसे पूछा जाता है कि why human RBC is the most preferred substance to study the composition or configuration of cell membrane, because mature human RBC is enucleated. Enucleated means nucleus is not present. enucleated होती है, न्यूक्लियस डिस्कार्ड हो जाता है मैच्योर ह्यूमन आरबीसी में. हाउएवर न्यूली फॉर्म्ड आरबीसी कंटेन्स न्यूक्लियस, पोजेस न्यूक्लियस, बट मैच्योर ह्यूमन आरबीसी enucleated in nature. Therefore, human RBC is the most preferred subject, most preferred substance to study the cell membrane. So, साइंटिस्ट स्टडी कर रहे थे, केमिकल स्टडीज कर रहे थे ह्यूमन आरबीसी पे. इनेबल्ड अ साइंटिस्ट टू डिड्यूस द पॉसिबल स्ट्रक्चर ऑफ प्लाज्मा मेम्ब्रेन, जिससे एक पॉसिबल स्ट्रक्चर प्लाज्मा मेम्ब्रेन का आइडिया लग रहा था. दिस स्टडी शोड दैट द सेल मेम्ब्रेन इज मेनली कंपोज्ड ऑफ लिपिड एंड प्रोटीन. और इन स्टडीज़ की सहायता से ये पता लग रहा था कि सेल मेम्ब्रेन मेनली किसकी बनी है, मेनली लिपिड और प्रोटीन की बनी है. द मेजर लिपिड्स आर फॉस्फोलिपिड, और उसमें भी द मेजर लिपिड, दिस इज द इम्पॉर्टेंट पॉइंट यू शुड नोट, द मेजर लिपिड्स आर फॉस्फोलिपिड दैट आर अरेंज इन अ बाइलेयर. सो ह्यूमन plasma membrane is mainly, plasma membrane or cell membrane is mainly composed of lipid and protein, and type of lipid that is present in plasma membrane or cell membrane, that is a phospholipid, the major lipid phospholipid, and that are arranged in bilayer, and there are two layers of phospholipids. Lipids are present in plasma membrane. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail towards the inner part. And in the arrangement of this lipid layer, one thing is you should know, that hydrophilic head is situated towards the outer surface, that is towards the aqueous environment, whereas hydrophobic tails are placed towards the inner side, away from the aqueous environment. So in case of lipid, you will find a hydrophilic head and hydrophobic tail. Hydrophilic head, hydro means loving, hydro means related to water, philic means loving, water loving, and hydrophobic, phobic means phobia, डरना किसी से, hydro water hating. So these tails are hydrophobic and this is the hydrophilic head. Lipid, in a molecule of lipid you will find a head. Head is made up of carboxylic group of fatty acid and hydrocarbon chain of fatty acid make the chain. So this is the hydrophilic head. Hydrophilic head is made up of carboxylic group. This is the carboxylic group of fatty acid that represents the hydrophilic head. And hydrophobic tails, these tails are basically hydrocarbon chains, C, 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 H, 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 hydrocarbon chains of fatty acid that form hydrophobic tail. So in case of lipid arrangement, the two layers of lipid that are arranged in such a manner that hydrophilic head is situated towards the aqueous environment, that is hydrophilic head facing extracellular fluid, and another lipid layer, the hydrophilic head is facing towards the cytosol or cytoplasm, and hydrophobic tails are towards the inner side, that is away from the aqueous solution. So the major lipid in case of cell membrane, major lipids are phospholipid that are arranged in a bilayer fashion. Also the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail towards the inner part. This ensures that the non-polar tail of saturated hydrocarbon, this type of arrangement makes sure that non-polar tail of saturated hydrocarbon is protected from the aqueous environment. As I told you earlier कि इस तरह का अरेंजमेंट है कि ये जो हाइड्रोफोबिक टेल्स हैं ये आपने अंदर की तरफ कर दी है, अवे फ्रॉम द एक्वस सिचुएशन, पानी से दूर कर दी है। पानी किधर है? तो ये आपकी लिपिड की बाई लेयर है, हाइड्रोफिलिक हेड है टूवर्ड्स द एक्वस कंडीशन। भाई ये आपकी प्लाज्मा मेम्ब्रेन है, तो प्लाज्मा मेम्ब्रेन के इधर इधर क्या है? प्लाज्मा मेम्ब्रेन के अंदर क्या होगा? या तो सेल का इंटरनल पार्ट ऑफ सेल साइटोप्लाज्म से भरा हुआ, यहाँ साइटोप्लाज्म है, फ्लूइड है, सेमी फ्लूइड मटेरियल, और सेल के बाहर क्या है? एक्स्ट्रा सेलुलर फ्लूइड, इंटरस्टिशियल स्पेस, और इस एक्स्ट्रा सेलुलर स्पेस में क्या होगा? एक्स्ट्रा सेलुलर फ्लूइड होगा, तो इधर भी फ्लूइड है, एक्स्ट्रा सेलुलर फ्लूइड है, और इधर साइटोप्लाज्म में। दोनों तरफ पानी है, आपने बचाना है अपनी टेल को पानी के संपर्क में हम नहीं आने देना चाहते। तो हाइड्रोफिलिक हेड इज टूवर्ड्स द एक्वस एनवायरनमेंट एंड हाइड्रोफोबिक टेल अवे फ्रॉम द एक्वस एनवायरनमेंट। सो हाइड्रोकार्बन, ये जो चेनें हैं हाइड्रोकार्बन, सैचुरेटेड हाइड्रोकार्बन की बनी, प्रोटेक्टेड फ्रॉम द एक्वस एनवायरनमेंट। इन एडिशन टू फॉस्फोलिपिड मेम्ब्रेन आल्सो कंटेन्स कोलेस्ट्रॉल। सो लिपिड कंटेंट जो इसमें है हमारे सेल मेम्ब्रेन में, मेनली तो फास्फोलिपिड है, बट केवल फास्फोलिपिड ही नहीं है, along with phospholipid cholesterol is also present. Cholesterol is also present. In addition to phospholipids, cell membrane also contains cholesterol. Later, biochemical investigations clearly revealed that the cell membrane also possesses protein and carbohydrate. तो पहले तो ये पता लगा कि भैया लिपिड की बनी बाई लेयर है, लेकिन बाद में इन्वेस्टिगेशन से, बायोकेमिकल इन्वेस्टिगेशन से ये पता लगा कि केवल लिपिड ही नहीं है, उसके अलावा प्रोटीन और कार्बोहाइड्रेट भी होते हैं सेल मेम्ब्रेन में। तो लेटर ऑन बायोकेमिकल इन्वेस्टिगेशन रिवील दैट only lipid is not the constituent of cell membrane, protein and carbohydrates are also present. The ratio of protein and lipid varies considerably in different cell type. और इसका रेशियो कितना होगा? आपको ये तो समझ में आ गया, ये वॉल है जो सीमेंट और मोरंग से बनी है, ठीक है, ये बात तो कंफर्म हो गई कि ये वॉल बनी है सीमेंट और मोरंग से। लेकिन कितना सीमेंट, कितना मोरंग, कितना लिपिड, कितना प्रोटीन, ये अलग अलग सेल में डिपेंड करता है। द रेशियो ऑफ प्रोटीन एंड लिपिड वेरीज कंसिडरेबली इन डिफरेंट सेल टाइप्स। इन ह्यूमन बींग्स द मेम्ब्रेन, ह्यूमन बींग में मेम्ब्रेन ऑफ द इरिथ्रोसाइट हैज़ अप्रॉक्सीमेटली 52% प्रोटीन एंड 40% लिपिड। सो ह्यूमन आरबीसी की जो मेम्ब्रेन है उसमें 52% प्रोटीन है एंड 40% लिपिड है। Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. And depending upon ease of extraction, अब आप इस लिपिड की लेयर से प्रोटीन को कितनी इजीली एक्सट्रैक्ट कर सकते हैं। Depending on the ease of extraction, membrane protein can be classified. Membrane protein को we have classified the membrane protein into two category, integral protein and peripheral protein, depending upon the ease of extraction. How easily you can extract the protein from that lipid bilayer. On behalf of that, we have classified the proteinous part protein into two categories, integral and peripheral. Peripheral proteins lie on the surface, peripheral or extrinsic protein that lie on the surface and easy to extract, while the integral proteins are partially or fully embedded in the, fully buried in the membrane, and these are difficult to extract. So there are two types of protein, two categories of protein, integral or peripheral. Peripheral proteins lie on the surface, whereas the integral proteins are partially or totally buried in membrane. So this is the diagrammatic representation of cell membrane. These are the hydrophilic head, this is also the hydrophilic head, these are the hydrophobic chains. So here you can see the presence of cholesterol along with phospholipid. Cholesterol is also present. These are the peripheral protein, extrinsic protein, which are easy to extract, and there are some integral proteins which are difficult to extract. They are partially or fully, these are the integral protein completely embedded in the lipid. phospholipid bilayer sugar. This is the sugar moiety attached to the protein, glycoprotein, peripheral protein, sugar is attached, and lipid, sugar is also attached. So this is the glycolipid and this is the glycoprotein. An improved model of structure of cell membrane was proposed by. So after all these findings, improved model of structure of cell membrane was proposed by Singer and Nicholson. So Singer and Nicholson model for the structure of cell membrane was given by Singer and Nicholson, scientists, in 1972, widely accepted as fluid mosaic model, and this model is known as fluid mosaic model. So you may get a question that fluid mosaic model was proposed by dash 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 scientist name and in the year dash dash dash. So Singer and Nicholson in the year 1972. Not only you have to remember the name of the scientist but also the year also. So Singer and Nicholson in 1972 widely accepted as fluid mosaic model. According to this model, the quasi-fluid nature, semi-fluid nature, lipid is semi-fluid in nature, not fully solid or not fully liquid. Quasi-fluid, semi-fluid. Semi-fluid nature of lipid enables lateral movement of protein within the overall bilayer. So because lipid is not solid, so it allows the movement of protein within the overall bilayer. This is the bilayer, lipid bilayer, and because lipid is not completely solid, it is semi-solid, semi-fluid, quasi-fluid, so it allows the movement of protein within the lipid bilayer. This ability to move within the membrane is measured as its fluidity, because the protein can move. Protein can change its position in the lipid bilayer. Protein can move in the lipid. This ability to move within the membrane measured as its fluidity. So as जितनी आसानी से प्रोटीन मूव हो सकेगी उतना ये फ्लूइड होगा. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation of intercellular junction, secretion, endocytosis, cell division. हम लोग बोल रहे हैं कि इसमें प्लाज्मा मेम्ब्रेन जो है, सेल मेम्ब्रेन जो है, क्वासी फ्लूइड लिपिड की बनी है और उसमें प्रोटीन मूव हो सकती है, जो उसकी फ्लूइड नेचर बता रहा है कि ये फ्लूइड नेचर है. तो फ्लूइड नेचर, इसको मेम्ब्रेन को कंसीडर करना कि इसका फ्लूइड नेचर है, लिपिड में प्रोटीन मूव हो सकती है, ये इस बात से भी इम्पॉर्टेंट है कि अगर ये हम लोग ये एज्यूम करें या ये मान के चलें कि प्रोटीन मूव हो सकती है लिपिड की लेयर में, जो उसके फ्लूइड नेचर को बता रहा, उसी बिहाफ पे हम इसके जो फंक्शन है प्लाज्मा मेम्ब्रेन के, सेल मेम्ब्रेन के, कि सेल ग्रोथ करती है. अरे भाई, अगर मानिए कोई ग्लास का कंटेनर, कोई स्टिफ चीज़ है, उसके अंदर कोई चीज़ आपने रख दी, तो ग्रो कैसे करेगी? ग्रो तभी करेगी जब ये बाहर की लेयर फ्लेक्सिबल हो, ये भी लचीली हो, ये भी उसके साथ ग्रो करे, रबर का जैसे बना कोई आर्टिकल. तो फ्लूइड नेचर, भाई सेल ग्रोथ कर रही है, उसका प्रोटोप्लाज्म वृद्धि कर रहा है, तो अगर ये इसकी बाहर वाली कवरिंग सेल मेम्ब्रेन साथ के साथ स्ट्रेच ना हो या इनलार्ज ना हो, तो कैसे कवर करे, कैसे वो गेन करेगा इंक्रीज इन डायमेंशन. सो आल्सो द पॉइंट ऑफ व्यू ऑफ फंक्शन लाइक सेल ग्रोथ करती है, तो फ्लूइड नेचर पे हम इसको एक्सप्लेन कर सकते हैं. फॉर्मेशन ऑफ इंटरसेलुलर जंक्शन, दो सेल्स के बीच में इंटरसेलुलर जंक्शन बनते हैं, उसको भी हम इसको फ्लूइड नेचर के आधार पे वी कैन एक्सप्लेन द formation of intercellular junction between two cells, keeping the fact in mind that this cell membrane is fluid in nature. Secretion, endocytosis and cell division can also be explained on this behalf. तो इस तरह से हम लोग देखते हैं कि फ्लूइड नेचर जो है, on the basis of fluid nature we can explain the different functions of cell membrane like cell growth, formation of intercellular junction, secretion, endocytosis, cell division, etc. Okay. One of the most important function of plasma membrane is the transport of molecule across it. This is the most important function. The most important function, one of the most important functions of plasma membrane is the transport of molecules across it. that it allows some molecules to get passed through it and does not allow some molecule to pass through it. The membrane is selectively permeable to some molecule present on either side of it. So there are 3 to 4 types of membrane. Fully permeable membrane does not form a barrier and each and every molecule can pass through it in both directions. This type of membrane is called as fully permeable. Impermeable membrane is that which does not allow any of the molecule to pass through it. That type of membrane is impermeable. And there are two terms, semi-permeable and selectively permeable. Semi-permeable membrane is that which allow the passage of molecule across it of solvent molecule only, not the solute. Solute molecules are not allowed. This type of membrane is called as semi-permeable. In case of semi-permeable membrane, only the passage of solvent molecules are allowed through the membrane. Solute is not allowed. And selectively permeable membrane is that which allows some of the solute particles, some of the solute molecules to pass through it. Selective solute molecules. So this type of membrane which allow some of the solute molecules to pass through it and doesn't allow other molecules to pass through it, that type of membrane is called selectively permeable. So here our plasma membrane or cell membrane is a type of selectively permeable membrane. One of the most important function of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side. Many molecules can move briefly across the membrane without any requirement of energy. Many molecules can move briefly across the membrane without involvement of energy, without requirement of energy, and this is called passive transport. This type of transport which do not require input of energy, which do not require the expenditure of energy, that type of movement is called passive movement or passive transport. Nutrient solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower concentration. What is diffusion? Diffusion is the movement of ion, atom or molecule of gases, liquid or solids from their high concentration area towards the lower concentration area without expenditure of energy. Down the gradient movement, down the hill. So when the molecules are moved from their high concentration area towards the lower concentration area without expenditure of energy, that is called as diffusion. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower concentration. Water may also move across this membrane from higher to lower concentration. Water means solvent. Movement of water by diffusion is called osmosis. So osmosis is the special type of diffusion in which the water molecule move from their high concentration area to lower concentration area. Osmosis is the movement of water molecule. Diffusion of water molecule is specific term. Osmosis is used for that. As the polar molecules cannot pass the non-polar lipid bilayer, polar molecules, those molecules which are carrying charge over their surface, those molecules are called polar molecules. Polar molecules find it difficult to cross the plasma membrane, non-polar lipid bilayer. They require a carrier protein. So their movement is facilitated by the carrier protein of the membrane to facilitate their transport across the membrane. A few ion or molecules are transported across the membrane. Few ion or molecules are transported across the membrane against their concentration gradient. Few ions and molecules are transported across the membrane against their concentration gradient from lower to higher concentration. Such a transport is a energy dependent transport, and in this process ATP is utilized and it is called active transport. So three types of transport are there. Passive transport. Passive transport is the movement of molecules or ion or atom from their high concentration area toward the lower concentration area without expenditure of energy. Passive transport. And when the polar substances try to move across the membrane, they require the presence of a specific protein, and their movement is facilitated by proteins. That proteins are called carrier protein or channel protein. So their movement, which is facilitated by the proteins, are called as facilitated movement. And facilitated movement is also occurs from high concentration area to the lower concentration area. Facilitated movement transport is not occur in the from the lower concentration to higher concentration. It is not occur uphill. Uphill transport is not accomplished by the passive movement. Only the active transport. In case of active transport only, the ion atom or molecules can move from their lower concentration toward the higher, that is uphill transport. Uphill transport can be accomplished only by the active transport with the expenditure of energy. Okay, so three types of movement: passive, facilitated and active transport are there. One of the most important function plasma membrane is the transport of molecules across it. तो चलिए हम लोग चलें उस पर, अपनी स्लाइड पे देख लें एक दो बातें. यहाँ तो सब बातें हम लोगों ने डिस्कस कर ली हैं. फ्लूइड मोज़ेक मॉडल और उसका अरेंजमेंट भी बाई लेयर का. 52% प्रोटीन, 40% लिपिड होता है. फास्फोलिपिड मेन है, उसके अलावा कोलेस्ट्रॉल मिल रहा है. हेड टूवर्ड्स द एक्वस पोजीशन है. ये सारी बातें जानने के बाद अब हम लोग देख लें कुछ एक्स्ट्रा हमें यहाँ से मिलता है. बिल्कुल मिलेगा. जो मिलेगा, फ्लूइडिटी क्या है? फ्लूइडिटी, हाउ इजली प्रोटीन कैन मूव विद इन द लिपिड बाइलेयर, दैट इज द फ्लूइडिटी. मूवमेंट ऑफ वाटर इज डिफ्यूजन, एक्वापोरिंग, इसकी ज़रूरत नहीं है डिस्कस करने की. लिपिड पार्ट इज क्वासिफ्लूइड. ये देखिए, ये वाली स्लाइड आप देखो, ये क्वेश्चन कभी कभी वो पूछ लेते हैं. तो इसमें आपके लिपिड की, ये लिपिड के हाइड्रोफिलिक हेड है, हाइड्रोफोबिक टेल है, हाइड्रोफिलिक हेड है, हाइड्रोफोबिक टेल है लिपिड. तो अगर लिपिड अपनी पोजीशन बदल के, इस पोजीशन से बदल के, आप देखिए इस पोजीशन पे आ गया सेकंड, तो फर्स्ट में ए और बी, ये इधर आ गया, ये इधर आ गया, पोजीशन चेंज हो गई. इसको हम लोग बोलेंगे लेटरल डिफ्यूज़न. लेटरल डिफ्यूज़न बोलेंगे, लेटरल और ट्रांजिशन, लेटरल मूवमेंट. प्रोटीन मॉलिक्यूल शो ओनली लेटरल और ट्रांजिशन मूवमेंट. इसको हम लोग फ्लिप फ्लॉप मूवमेंट नहीं बोलेंगे. फ्लिप फ्लॉप मूवमेंट कभी कभी वो पूछ देते हैं आपसे, हालांकि अब पूछा ना जाएगा क्योंकि अपनी एनसीईआरटी में मेंशन नहीं है. तो प्रोटीन केवल इस तरह से ट्रांसवर्स मूवमेंट कर सकता है. प्रोटीन ओनली ट्रांसवर्स मूवमेंट कर सकता है. यहाँ पे लिपिड मॉलिक्यूल को दिखाया, लेकिन प्रोटीन मॉलिक्यूल भी ट्रांसवर्स मूवमेंट कर सकते हैं. प्रोटीन मॉलिक्यूल शो ओनली ट्रांजिशन, प्रोटीन मॉलिक्यूल ओनली लेटरल मूवमेंट कर सकते हैं. सॉरी, मैं बोल रहा था ट्रांसवर्स. प्रोटीन मॉलिक्यूल ओनली, फिर से देख लीजिए, समझ लीजिए, ये वाला लेटरल मूवमेंट है और ये वाला ट्रांसवर्स मूवमेंट है कि यहाँ वाला यहाँ आ जाए. तो ये जो ये स्लाइड है, ये आपकी ऊपर वाली, ये ट्रांसवर्स मूवमेंट दिखा रही है जो लिपिड कर सकता है, और ये वाला लेटरल मूवमेंट इस तरह से. लेटरल मूवमेंट आपका लिपिड भी कर सकता है, प्रोटीन भी कर सकता है. ट्रांसवर्स मूवमेंट ओनली लिपिड कैन डू. प्रोटीन मॉलिक्यूल डू नॉट शो ट्रांसवर्स मूवमेंट. और ये ट्रांसवर्स मूवमेंट ही आपका फ्लिप फ्लॉप मूवमेंट है. तो फ्लिप फ्लॉप मूवमेंट कौन दिखा सकता है? लिपिड दिखा सकता है. ठीक है, फिर से एक बार 2 मिनट समझिए, सब बातें इरेज कर दीजिए. ये हमारा लिपिड की बाई लेयर है, ये हाइड्रोफिलिक हेड, ये हाइड्रोफोबिक टेल. अगर ये लिपिड का मॉलिक्यूल अपनी पोजीशन बदल के इस पोजीशन से इस पोजीशन पे आ जाए, तो ये ट्रांसवर्स मूवमेंट है, ये फ्लिप फ्लॉप मूवमेंट कहलाता है. और अगर ये मॉलिक्यूल अपना पोजीशन बदल के इस पोजीशन पे आ जाए, तो ये लेटरल मूवमेंट है. लिपिड का मॉलिक्यूल लेटरल मूवमेंट भी दिखाता है, ट्रांसवर्स भी. ट्रांसवर्स मूवमेंट को हम लोग फ्लिप फ्लॉप मूवमेंट कहते हैं, और लेटरल मूवमेंट को हम लोग ट्रांजिशन मूवमेंट बोलते हैं. ये प्रोटीन, ओनली प्रोटीन प्रोटीन केवल इसी तरह का मूवमेंट दिखा सकता है, लेटरल प्रोटीन ट्रांसवर्स मूवमेंट नहीं दिखाता। अब हम लोग देख लें पॉइंट जो हम लोगों ने डिस्कस किए, कोई पॉइंट ऐसा जो रह गया हो। चलिए चलें वापस उधर। तो ये बात हो गई आपकी सेल मेम्ब्रेन की, ठीक है जी। सेल मेम्ब्रेन में आपने देखा लिपिड की बनी बाई लेयर है जिसमें प्रोटीन एम्बेडेड है। प्रोटीन on the basis of ease of extraction can be of two type, peripheral protein or integral protein. Peripheral proteins are placed or situated on the outer side and can be easily extracted, whereas integral proteins are partially or completely embedded, buried in the protein bilayer or lipid bilayer, so it is difficult to extract them. ये बात हो गई आपकी सेल मेम्ब्रेन की। और सेल मेम्ब्रेन में आप लोग को 1972 में इसको सिंगर एंड निकोलसन ने एक्सप्लेन किया है। इसके स्ट्रक्चर को फ्लूइड मोज़ेक मॉडल कहलाता है। लिपिड की बनी बाई लेयर है और इसमें प्रोटीन और कार्बोहाइड्रेट भी है। ओके, अब हम लोग आ जाएं सेल वॉल पे। मूव टू द नेक्स्ट, सेल वॉल। एज यू मे रिकॉल, जैसा कि आप याद कर सकते हैं, नॉन लिविंग रिजिड स्ट्रक्चर कॉल्ड द सेल वॉल फॉर्म्स एन आउटर कवरिंग फॉर द प्लाज्मा मेम्ब्रेन ऑफ फंजाई एंड प्लांट। अगर आपको याद हो, फंजाई और प्लांट में आउटर मोस्ट बाउंड्री जो है सेल की कौन बना रहा है? सेल वॉल बना रहा है। सेल वॉल नॉट ओनली गिव शेप टू द सेल बट प्रोटेक्ट द सेल फ्रॉम मैकेनिकल डैमेज एंड इन्फेक्शन। सेल वॉल का काम क्या है? इट प्रोटेक्ट्स द सेल फ्रॉम मैकेनिकल डैमेज एंड इन्फेक्शन एंड आल्सो गिव शेप टू द सेल। इट आल्सो हेल्प्स इन सेल टू सेल इंटरेक्शन। साथ ही साथ सेल वॉल का फंक्शन है सेल टू सेल इंटरेक्शन में हेल्प कर रहा है एंड प्रोवाइड्स बैरियर टू अनडिजायरेबल मैक्रोमोलिक्यूल। साथ में बैरियर बना रहा अनडिजायरेबल मैक्रोमोलिक्यूल के लिए। सेल टू सेल इंटरेक्शन को प्रमोट कर रहा है, सेल टू सेल इंटरेक्शन को प्रमोट कर रहा है, मैकेनिकल डैमेज से बचा रहा, इन्फेक्शन से बचा रहा एंड प्रोवाइड्स बैरियर टू अनडिजायरेबल, और बैरियर बना रहा अनडिजायरेबल मैक्रोमोलिक्यूल के लिए। बैरियर बना रहा अनडिजायरेबल मैक्रोमोलिक्यूल के लिए, बैरियर बना रहा। एल्गी हैव सेल वॉल मेड अप ऑफ सेलुलोज, ग्लेक्टेंस, मैनेंस। एल्गा में सेल वॉल का कंपोजिशन याद कर लीजिएगा। एल्गल सेल वॉल इज मेड अप ऑफ सेलुलोज, ग्लेक्टेंस, मैनेंस। और मिनरल्स मिलेंगे लाइक कैल्शियम कार्बोनेट। While plants, it consists of cellulose, hemicellulose, pectins, proteins. Cellulose, hemicellulose, pectins, proteins. This is the composition of plant cell. Whereas the plant cell wall, whereas the algal cell wall is made up of cellulose, galactans, mannans. Cellulose, galactans, mannans, and minerals like calcium carbonate. So this is the important. आपको ये याद होना चाहिए क्योंकि फंगल सेल वॉल इज मेड अप ऑफ काइटिन। तो इन तीनों के आपको याद होना चाहिए। एल्गल सेल वॉल सेलुलोज, एल्गल सेल वॉल इज मेड अप ऑफ सेलुलोज, गैलेक्टेंस, मैनेंस एंड कैल्शियम कार्बोनेट, वेयरएज प्लांट सेल वॉल इज मेड अप ऑफ सेलुलोज, हेमीसेलुलोज, पेक्टिन्स एंड प्रोटीन्स। द सेल वॉल ऑफ अ यंग प्लांट सेल, द सेल वॉल ऑफ अ यंग प्लांट सेल, द प्राइमरी वॉल इज केपेबल ऑफ ग्रोथ। प्राइमरी सेल वॉल केपेबल होती है ग्रोथ के, which gradually diminishes as the cell matures and the secondary wall is formed on the inner side of the cell। और ये सेल वॉल जो प्राइमरी सेल वॉल है, इसकी ये जो कैपेसिटी है दैट इट कैन ग्रो, ये धीरे 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 जैसे जैसे सेल मैच्योर होती है इसकी कैपेसिटी ख़त्म होती चली जाती है। एंड एट द मैच्योरिटी ऑफ द सेल, सेल जब फुल्ली मैच्योर हो जाती है तब प्राइमरी सेल वॉल के अंदर सेकेंडरी सेल वॉल का डिपोजिशन होता है। So primary cell wall, then the second layer, secondary cell wall is formed. Secondary cell wall is laid down over the surface of primary cell wall. Secondary cell wall is formed on the inner towards membrane side of cell. The middle lamella is layer mainly of calcium pectate which holds or glues the different neighboring cells together. Middle lamella is the cementing agent. It acts as a cementing agent. It acts as a glue between two adjacent cells. So middle lamella is a layer mainly of calcium pectate, and middle lamella is made up of mainly of calcium pectate and magnesium pectate. Middle lamella mainly made up of calcium pectate and magnesium pectate, and it acts as a glue between two adjacent cells, which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by the plasmodesmata. The cell wall, middle lamella may be traversed by plasmodesmata, which connect the cytoplasm of two neighboring cells. So cytoplasm of two neighboring cells are in contact with each other by means of plasmodesmata, cytoplasmic bridge. That is the cytoplasmic bridge. So this is about few lines about middle lamella to cell wall, middle lamella. Now move to the next segment, that is the endomembrane system. So prior to moving the next segment, endomembrane system, we should discuss some question about cell wall and cell membrane. So algal cell wall, as your NCERT says, algal cell wall is made up of cellulose, galactans, mannans and minerals like calcium carbonate, while plant cell wall, cellulose, hemicellulose, pectin and proteins. Fungal cell wall is made up of chitin. Chitin is an example of carbohydrate and modified form of cellulose. This is the important. Please note these lines. Fungal cell wall is made up of chitin, that is the polymer of N-acetyl glucosamine. It is an example of carbohydrate. Chitin is a complex carbohydrate and modified form of cellulose. Cellulose का modified form है. Eukaryotic cell. Topic 2. Plant cell differs from animal cell in having plastid, cell wall, large central vacuole, all of these. You should go with all of these. This is the difference between plant cell and animal cell. The best material for the study of the structure of cell membrane or composition of cell membrane, RBC of human, as I told you earlier, because RBC of human, mature human RBC is enucleated, nucleus is not present. Which chemical property is shared by all types of lipid forming the plasma membrane? Sugar component, glycerol, phosphate, hydrophobic reason. All types of lipid that form the plasma membrane contains hydrophilic head and hydrophobic region. Presence of hydrophobic region is the characteristic feature. A phospholipid molecule is amphipathic and arranged in two layers. फास्फोलिपिड दो लेयर में अरेंज है. एम्फीपैथिक है कि उसमें हाइड्रोफिलिक हेड भी है, हाइड्रोफोबिक टेल भी है. द हेड ऑफ हाइड्रोफिलिक मॉलिक्यूल इज प्रेजेंट टुवर्ड्स द एक्वस मीडियम, away from the aqueous medium, present anywhere, embedded in protein. So head is present towards the aqueous medium, hydrophilic, water loving. This is facing the aqueous condition, either the extracellular fluid or cytoplasm. So head is the head of the phospholipid molecule is present towards the aqueous medium. Most abundant lipid in the cell membrane is phospholipid. However, cholesterol is also present. Cell recognition. So select the incorrect statement. Ratio of protein and lipid vary considerably. Yes, this is correct. 52% protein and 40% lipid constitute the membrane of human RBC. That is correct. Tail of lipid is hydrophobic and is in contact with cytoplasm. Tail of lipid is hydrophobic. Up to that much it is correct, but it is not contact. It is away from cytoplasm. So tail is towards the inner side, away from the head of lipid is hydrophilic. Yes, this is also correct. This statement is not correct. Hydrophobic tail is away from the cytoplasm. Integral cell membrane proteins. Integral cell membrane proteins are partially embedded, hard to remove, show only lateral movement but not vertical. Lateral movement show करेगी, flip flop movement show नहीं करेगी. All of this you should go with all of these. Lipids are arranged within the membrane with polar heads towards inner side and hydrophobic tail towards outside. No, the tail is polar hydrocarbon. It exposed to aqueous. No, not exposed to aqueous. The hydrophobic tail of lipid being of inner side ensure their protection from aqueous. Yes, this is the correct. Lipids are arranged within the membrane with the hydrophobic tail being on the inner side, and this ensures that they get protected from the aqueous environment. tail is hydrophilic, tail is hydrophobic. So these three are not the correct statement. You should opt option number C is the correct. The fluid mosaic model explains structural aspect. It also explains fluidity, the secretion, repairing, growth, all these functions. Endocytosis can be explained on the basis of fluid mosaic model, both structural and functional, only fluidity. So you should go with both structural functional aspect of cell membrane. It can explain both structural and functional. Fluid nature of membrane is able to explain. हो गई बात, चलिए आगे. Cell membrane is selectively permeable. We know that cell membrane is selectively permeable. This means that allow all material, that is the type of fully permeable, which the membrane which allow all materials to pass through it, that is known as fully permeable. Allow only water, only solute pot, only solvent can move, that is called as semi permeable. Allow only certain, yes, allow only certain materials to pass through it, that is called as selectively permeable. Which of the following is not correct regarding cell membrane? Generally, smaller molecule pass easily without energy requirement. Water soluble substances pass through the substances. Neutral solute move across it by simple diffusion. None of the above. Water soluble substances pass through the substances, pass through the membrane. So all are correct. Which of the following is not a constituent of cell membrane? Cell membrane का constituent क्यों नहीं है? प्रोलीन नहीं है, ग्लाइकोलिपिड होते हैं, फास्फोलिपिड होते हैं, कोलेस्ट्रॉल होते हैं, प्रोलीन नहीं होता. Which of the following is primary lipid in cell membrane? Mainly lipid कौन सा है? प्रीडोमिनेंट लिपिड हमारा फास्फोलिपिड है. What is the false about osmosis? Specific form of diffusion, correct है. Refers to the movement of water along the concentration gradient, correct है. It is passive movement of water, correct है. It occurs through a carrier protein and needs ATP. This is not the correct statement about osmosis. Which of the following characteristic is correct about the plant cell wall? Play role in protection, helps in cell to cell interaction, provides a barrier. All are correct. You should go with all of the above. Composition of plant cell. Plant cell किसका बना हुआ है? Cellulose, hemicellulose, pectin and protein. Galactans. Galactans are, you will find the galactans in algal cell wall. Which of the following is correct regarding middle lamella? It is formed as a cell plate during cytokinesis, mainly composed of calcium carbonate, holds glues together of two different cells, two neighboring cells. All of the above are correct regarding middle lamella, formed as a cell plate during cytokinesis. Phospholipid molecule has a head and two tails. The tails are found in the interior of the membrane. Tails are found in the interior side. At the surface of membrane? No, and not at the surface of membrane. Tails are basically remain away from the aqueous condition, that is towards the inner side. The fluid mosaic model was given by Singer and Nicholson in the year 1972. Singer and Nicholson model, the extrinsic proteins are situated towards the periphery and are easy to extract. Loosely associated but cannot be easily tightly associated. Loosely associated with intrinsic protein and can be easily separated. The latest model of cell membrane is fluid mosaic model. So that finishes with cell wall and cell membrane questionnaire. Now we will move to the endomembrane system. The next topic is endomembrane system. So we should discuss this topic in the next lecture. यहाँ पे आके मैं रोकता हूँ और नेक्स्ट लेक्चर में मैं एंडोमेम्ब्रेन सिस्टम को डिस्कस करता हूँ, क्योंकि ऑलरेडी 50 मिनट की वीडियो हो गई है. आप लोग हो जाओगे बोर। तो इसके आप लगा लो क्वेश्चन यहाँ पे आके। अब मैं अपनी वाणी को विराम देता हूँ। मिलते हैं आपसे ब्रेक के बाद नेक्स्ट लेक्चर में रिगार्डिंग एंडोमेम्ब्रेन सिस्टम। इन द नेक्स्ट लेक्चर वी आर गोइंग टू डिस्कस एंडोमेम्ब्रेन सिस्टम। तो क्वेश्चन जरूर लगा लीजिए, क्वेश्चन लगवाए भी हैं। मिलते हैं आपसे ब्रेक के बाद। हैव अ नाइस डे। मिलता हूँ ब्रेक के बाद। टिल देन गुड लक, गुड बाय, थैंक यू। अगर अच्छा लगा हो लेक्चर लाइक कर दीजिए, चैनल सब्सक्राइब कर लीजिए और आइए अगली क्लास में आपके इंतज़ार में।